Hi, let's study about Pureline selection. So Pureline selection is a type of selection method which is used in plant breeding. You know the other two selection methods usually adopted like mass selection and clonal selection. So first of all, what is meant by a Pureline? Pureline is a homozygous individual or a pure individual. How is it obtained? It is obtained from the parental plants by continuously self-pollinating them. So, it's a Pureline is a homozygous individual. You know what is meant by a homozygous individual or homozygosity? In genetics, you might have studied the genotypes capital T, small t, etc. regarding tallness. So, that is a homozygous nature. So, likewise, the Pureline is a homozygous individual that is produced by continuous self-pollination of their parents. Okay, so these are the Pureline progenies that we have to get. So what's the importance of these Purelines? The Purelines can be used for introducing a new variety as in plant breeding. Then these can be used as parents in hybridization methods, and also these Purelines can be used for studying mutation, because normally in Purelines, this Pureline selection method, or in the case of Purelines, chance of mutation or variation is much less. Maybe some accidental method, by some accidental methods, mutation can be effected. So these Purelines can be used for studying the mutation also, mutation in plants. So that's the importance of Purelines. Now coming to the Pureline selection method. In Pureline selection method, individual plants are selected from a group of plants, and the Purelines are or pure progenies are produced by continuous self-pollination. Okay, so this Pureline selection method is a tedious process, and it takes about 10 years for the release of final cultivar varieties. So in the first year, so different steps are there in Pureline selection. So in the first year, what we do is, or what the farmers do is, individual plants having desirable qualities are selected from the group of plants. Maybe some fruit size, fruit color, flower color, or whatever it is. So individual desirable characters are considered and individual plants are selected. In the next year, what the farmers do is, they are individually grown without getting mixed up, right? If there is one plant I have selected, that plant is grown at a particular place, and the second plant is grown far away from the first plant. So the individuality is maintained in the Pureline selection method, right? So in the coming year, what is done is, self-pollination is done in these plants individually, and the seeds are collected individually or separately from each plant. So not that the individuality is very important in this case in Pureline selection. Okay, so this step can be continued for 2 or 3 years also. Anyway, then field trials are done. Field trials means the farmers compare the quality of these plants with the available cultivar varieties, already available ones. And again, in every step, the plants having the best qualities are selected, and continuously the process is repeated, the process is repeated till 7 years. Okay, so after the 7th year, the cultivars or these varieties are released for seed testing and seed certification. In India, all these things are done by ICAR, Indian Council of Agricultural Research. And the seed certification and seed testing and approval, everything is done based on the rules of All India Cooperative Crop Improvement Project. Okay, so after testing and all, again in ICAR, the seeds are again conducted for, or seeds are again crop tested or field tested, and after the 10th year only the final cultivars are released to the farmers. So it's a tedious, time-consuming process, and it takes about 10 years for the final release of cultivars. Okay, so large number of plants like wheat, rice, then cotton, etc. We are having large number of varieties, new cultivars, which were produced by this Pureline selection method. So once again, Pureline selection method is a selection method where individual plants are selected from a group of individuals or group of plants, and the pure lines are obtained by continuous self-pollination of these plants. Hope you have understood. Thank you.